Why gene regulation matters. No organism uses all its genes all the time. Instead, cells turn genes on and off depending on the situation. The ability to control or regulate genes, therefore, underlies all of life's processes such as growth, metabolism, and the ability to adapt to a changing environment. In US, breast cancer is the second biggest killer cancer in women after lung cancer. It is seen, research has shown that if the breast cancer is detected very early on, in the early stages, and it is treated, then the chances of the patient surviving 5 years or more is 10%, 100%. However, if the breast cancer is not detected and treated until it has metastasized, that means it has spread throughout the body, the 5-year survival rate is below 25%. So what is the treatment regimen? What does it look like? It looks like, first of all, it involves surgery. Then this is followed by hormone therapy. Then the patient is given radiation to kill the cancer cells. And finally, if there are still some cancer cells surviving, then chemotherapy is used. How do specialists know which cancers are risky and they require chemotherapy? Usually a genetic analysis is done because most of the times cancers are produced by mutations in genes. So when the genes that get mutated, if the tumor suppressor genes have been turned off and the oncogenes have been turned on, then we know that those cells are going to become cancerous. So scientists can analyze which genes have mutated and what are the repercussions, what are the effects. So if the genes of all the cancer patients, can be detected, in future, then we will know which patients require a more aggressive treatment than others. These cells from a cancerous cervical tumor have lost the ability to control their growth. That means they do not have any contact inhibition. How and why genes are regulated. To understand this process, we have to start from the very beginning of life. Life starts when a sperm fertilizes an ovum and a fertilized egg or a zygote is produced. That's a single cell. And that single cell divides rapidly by mitosis, produces two cells from 2 to 4, 4 to 8, 8 to 16, and so on. So every cell in your body has the same DNA, that means the same genetic material, as that first cell in the zygote, and every somatic cell would contain every single gene. But we see that the cells in our body, they have different functions. They are specialized in their structure and function. Our heart cells function in a different way than our neurons, that means the nerve cells. So how does this happen? This happens because of specialization, a process called differentiation. That means all of these cells might have the same genetic information, but the cells with the same genetic information can develop into different types of cells through the process of gene control or gene regulation. That means mechanisms that turn on certain genes while other genes remain turned off. So controlling the gene activity allows for specialization of cells within the body. The various types of cells in any multicellular organism, they owe their distinctiveness to different combinations of genes being turned on and off by gene regulation in each cell type. So what does it mean that a gene is turned on or off? So what is a gene? It's a segment of DNA, right? A sequence of nucleotides. And then from that DNA, remember, mRNA is made. And then the mRNA gets translated into protein. So this is this gene flow, information, or the central dogma, DNA to mRNA to protein. A gene that is turned on means that is the gene that is being transcribed and it will be translated into specific proteins. And if a gene is turned off, that means that part of DNA is not going to be transcribed into mRNA and therefore will not be translated into a protein. So this overall process by which genetic information flows from DNA or genes to the proteins is called gene expression. The control of gene expression makes it possible for cells to produce specific kinds of proteins when and where they are needed, allowing cells to respond quickly and efficiently to information from the environment. This figure shows the patterns of gene expression in three different types of human cells. You see that different types of cells express different combinations of genes. Let's start with gene regulation in bacteria. Let us consider a bacterium living in your intestines, and that bacterium is E. coli or Escherichia coli. What happens when you drink milk products, such as a milkshake? So suddenly your body, your digestive system has taken it in and it has broken it down into simpler sugars. And one of those sugars is lactose. So when the lactose level rises, in response, what does E. coli bacterium, it does is it will express genes, few genes that are specific, that are going to transcribe and then translate enzymes, means proteins, that will enable the bacteria to absorb and digest this sugar. So when the level of lactose is high, these particular enzymes will be turned on, protein will be made, enzymes will digest and absorb the lactose until it is gone. And when the lactose level falls down, these genes are going to be turned off. This is how homeostasis, that means your body surviving in or liking an optimal range of conditions, this is how it is maintained. Okay, this is how metabolism is maintained in your body. How does the presence of lactose in your diet control the activity of genes? It's because these genes are arranged in such a way in the DNA that they behave as a single unit. They are, placed right next to each other. They are adjacent and they get turned on and off as a single unit. And such a cluster of related genes and sequences, okay, that are being regulated, being controlled at the same time, this particular cluster is known as an operon. The operon considered here is the lac operon, short for lactose. So are there any specific control sequences in DNA that will regulate other genes? Yes, there are. And how do these DNA control sequences turn genes on or off? Well, one such regulator or control sequence called a promoter is the site where a particular enzyme, the RNA polymerase, attaches and it starts transcription. Between this promoter part and the protein producing or enzyme genes, there would be a DNA segment, which is known as the operator part. This acts as a switch that is turned on or off, depending on whether the specific enzyme or a protein is bound there. So the operator and the protein together determine whether RNA polymerase can attach to the promoter and start the process of transcription. To summarize what we've learned about gene regulation in bacteria so far, an operon is a cluster of genes with related functions together with a promoter and other non-protein coding DNA sequences that control their transcription. For example, the lac operon allows E. coli to produce enzymes for lactose only when the sugar is present. Now let us look at the figure in the next slide. The top half of this figure shows the lac operon in off mode, its status when there is no lactose available. Transcription is turned off because first, a protein called a repressor, that is shown in red color, binds to the operator shown in yellow color and physically blocks the attachment of the RNA polymerase to the promoter. The bottom half of this figure shows the operon in the on mode when lactose is present. The lactose interferes with the attachment of the lac repressor to the operator by, number first, you see binding to the repressor, and number two, changing the repressor's shape. As we so often see in biological systems, structure and function are related. In this case, altering the repressor's shape changes how it acts. In its new shape, the repressor cannot bind to the operator and the operator switch remains on. Now, number three, RNA polymerase is no longer blocked, so it can now bind to the promoter and from there, number four, transcribe the genes for the lactose enzyme into mRNA. Number fifth, translation produces all three lactose enzymes shown in purple. We just learned about this in the previous slide. Mechanisms of gene regulation in eukaryotes, particularly multicellular eukaryotes, are much more specialized and sophisticated than the bacteria. So the flow from gene to protein in eukaryotic cells is a long one. So we have several checkpoints at which genes can be turned on, and the process can be turned on or off. And we use a particular analogy to explain this, as you will see in the next slide. Picture the series of pipes that carry water from your local reservoir to a faucet in your home. At various points, valves control the flow of water. We use this analogy in the figure here to illustrate the flow of genetic information from a eukaryotic chromosome, which is a reservoir of genetic information, to an active protein that has been made in the cell's cytoplasm. The multiple mechanisms that control gene expression are analogous to the control valves in your water pipes. In the figure, each control knob indicates a gene expression valve. All these knobs represent possible control units, although only one or a few control points are likely to be important for a typical protein. So we'll use a reduced version of this figure as a guide to explore several ways that eukaryotes can control gene expression starting within the nucleus. DNA packing. DNA packing tends to block gene expression by preventing access of transcription proteins to the DNA. An extreme example is X chromosome inactivation in the cells of female mammals. X chromosome inactivation is seen in female mammals, where one X chromosome in each somatic cell gets highly compacted and almost entirely inactive. This X chromosome inactivation first takes place early in the embryonic development, when one of the two X chromosomes in each cell is inactivated at random. So after one X chromosome is inactivated in each embryonic cell, all of that cell's descendants will have the same X chromosome turned off. Consequently, if one X chromosome in the embryonic cell has one allele and the other has a different allele, about half of the cell's descendants will express one allele, while the other half will express the other allele. You see this case of tortoise shell pattern on a female cat here. This phenotype requires two different alleles, an allele for orange fur on one X chromosome and an allele for black fur on the other X chromosome. Orange patches are formed by a population of cells in which the X chromosome with the allele for orange fur is active. Black patches have cells in which the X chromosome with the allele for black fur is active. The most important control points, called transcription factors, interact with DNA sequences called enhancers and the promoter, and also transcription factors interact with each other to turn the transcription of eukaryotic genes. This is a model for turning on. Model for turning on eukaryotic gene, and we'll learn about this in the next slide with the animation.
The fine-tuning of gene expression copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene. This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene. The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase to initiate transcription.
So there is an existence of two types of proteins, one that turns on transcription and the other that turns off transcription. The ones that switches on transcription are known as activators, and the one that turn off the transcription are called the repressor protein. Now, the default state for most genes in multicellular organisms is they seem to be always kept, off. So they will be turned on only when required. There are certain exceptions like housekeeping genes for routine maintenance activities of the body which stay on. There are also opportunities for the control of eukaryotic gene expression after transcription when introns are cut off the RNA and a cap and tail are added to process RNA transcripts into mRNA. Within a cell, RNA splicing, that means the splicing together of exons, which is a part of post-transcription RNA processing, this can occur in more than one way. lead to the formation of types of mRNA molecules in the same starting the RNAs, but this RNA breakdown provides another opportunity for gene regulation. This figure shows how multiple types of mRNAs can be produced from the same gene using alternative RNA splicing.
A cell can rapidly adjust to environmental changes by regulating several processes that happen after transcription. In RNA processing, a cap and tail are added, introns removed, and the remaining exons spliced together. Alternative splicing of RNA may create different mRNA molecules from the same primary RNA transcript.
The lifespan of mRNA molecules helps determine how much of a protein is made. Nucleotide sequences that affect mRNA longevity are often found in the 3' untranslated region following the stop codon. Enzymes attack the poly-A tail first, and eventually the rest of the mRNA. Depending on its ability to resist this degradation, a eukaryotic mRNA molecule may last from hours to weeks. The longer an mRNA lasts, the more protein is made.
In the cytoplasm, presence of microRNAs and small interfering RNAs may block the translation of an mRNA, and various proteins may regulate the start of translation. So the microRNAs are small single-stranded RNA molecules which bind to complementary sequences on mRNA molecules. In addition to this, there is another class of small RNA molecules called small interfering RNAs, siRNAs, and these block gene expression. This blocking is known as RNA interference or RNAi. The figure shows how a microRNA regulates gene expression. Finally, more gene regulation may occur at the last step of gene flow, which is translation, in which an mRNA is used to make a protein. So the cell may activate the finished protein in various ways, for instance, by cutting out portions or chemical modifications. Eventually, the protein may be selectively broken down.
Often, polypeptides must be modified in some way to produce functional proteins. For example, a polypeptide is cleaved in 2 or more polypeptide chains. Other molecules might be attached to the protein, or the protein might be phosphorylated. Some proteins need to be transported to specific destinations. A cell can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tagged protein.
Cell signaling means cell communication. A multicellular organism contains multiple cells. One cell needs to chemically communicate with another cell, and sometimes it might take a series of events for one cell to talk to another and then an effect to occur. And this entire pathway is known as signal transduction pathway. This figure shows how a signal molecule from one cell alters the gene expression in a target cell without entering the target cell.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function